Hi, so this is the lesson that we missed from the Friday before vacation due to our assembly. I'm pretty sure that we went through the warm-up questions here, but if you wanted to take a look just to make sure you're up to date on those, that'd be great. I also reminded you that the claim evidence reasoning and some pictures for the results of your DNA extraction lab will be collected on Monday, April 23rd. Please make sure that any images you include are original images and that you have them properly captioned and include some labels as well. I definitely want you to use simplicity in your claim and be very specific about what evidence there is to support your claim. The lesson itself that we missed, or we got started with anyway, is all about DNA replication. We know that DNA replication is a really important precursor to mitosis and meiosis. In order for there to be enough genetic information to support the production of new cells, there first has to be a stage where that amplification of DNA happens. And so as we noted in class on Friday, this image is showing mitosis, the stages of mitosis. Of course, the cell that would precede prophase would be the parent cell that is in undergoing interphase, and so it's during S phase specifically of interphase where DNA replication happens. This is facilitated obviously by the structure of DNA, and as Watson and Crick showed, DNA has these, sort of rungs in the middle of this ladder that are pairings of A to T's and G to C's. So these nitrogen bases will hydrogen bond in the middle, while the backbone, the phosphate, and the sugar will be sort of repeating units. Nucleotides are the basic fundamental subunit of DNA, and remember that DNA runs anti-parallel. So as one strand is going 5' to 3', the other strand is moving in the opposite direction. And we'll be able to see that in an image later on in the lesson. There are a lot of enzymes and there's a lot going on during S phase, but I wanted to point out the most important features of DNA replication, and that is, number one, the first thing that has to happen is that the DNA strands themselves have to be pulled apart, and so this is accomplished by means of DNA helicase. It's an enzyme that will break through those hydrogen bonds that are holding those nitrogen bases together. Once those sort of strands have been exposed, now you have these sort of what is referred to as replication bubbles that are going to be forming, and it is because of that we now have these exposed strands. Before DNA polymerase III, which is a major and principal enzyme involved in DNA replication, can do its job, which is to deliver free DNA nucleotides to each complementary strand, each strand has to be primed. Just like you put primer on a wall before you paint it, primase is necessary in order to deliver free complementary RNA nucleotides to the beginning of each strand so that DNA polymerase III has something to bind to. So again, DNA polymerase III cannot start from scratch. It needs a primer, an RNA primer, to initiate that. Once that has been accomplished, DNA polymerase I can go back and replace those RNA nucleotides with DNA nucleotide complements, and then any gaps in that, in those structures can be sealed up with ligase. Here's an image of DNA replication, just a schematic of it. You'll see that the blue strands represent the 5' to 3' and 3' to 5' of that parent strand. You'll also notice that in order to make sure that anti-parallelism is, continues, we need to be building in opposite directions here. So as one strand is being built from 5 to 3 on the left, that red strand, the other strand, a complementary strand to that parent, is being built in the opposite direction. And as you can see, fragments are going to be forming on that strand on the right, which are called Okazaki fragments, from the Japanese scientist who discovered those fragments, and we refer to that as being sort of discontinuous. So while the red strand on the left is continuously being built in the direction of helicase, the strand on the left is being built discontinuously. And so this is referred to as the lagging strand. So we have the leading and the lagging strand. In my other classes, we had a chance to actually build this, so I'm going to go through how I demonstrated that during class, how everybody in class had an opportunity to do that. So I have a couple of models that I will show you. So we first started using these foam pieces to build nucleotides, and this again is a model. The green thing sticking out towards you is a guanine. I think it's one of the nitrogen bases, but it has been inserted into the sugar part of our nucleotide nucleotide. So remember, it's the nitrogen base that is attached to a sugar, deoxyribose, which is attached to phosphate. It's that circular end that is the phosphate end, which is consistent with the 5' end of DNA. Once you start to build this model, you can see we're building from 5' to 3'. So the next picture you should see are a couple of these nucleotides that I have just sort of connected to each other. And we connected these in a very specific sequence, and I think the sequence is shown in a previous slide. I didn't actually follow that, but you kind of get the idea here. So in order to build, so I just added more nucleotides. Remember, we build with nucleotides. We do not build a backbone and then stick in those nitrogen bases. That was the purpose of that marshmallow toothpick exercise. So once we have a complete strand, then you can build from in the opposite direction, that complementary strand. And so you'll see that there are that these pieces, the red and the blue and the green and the yellow pieces, are going to be able to fit together very nicely with complementary nitrogen bases. So again, this is building the parent strand that will eventually undergo DNA replication during interphase. And so as you can see here, the green nucleotide has the 5' end, and then the blue nucleotide is facing in the opposite direction. So the blue is actually going to be your 3' end. And so this is kind of how I initially built that the parent DNA strand. And so it's this parent DNA strand, as red and yellow are complements and blue and green are complements, that will effectively be copied. We refer to DNA replication as being semi-conservative because the end products will always have a strand from the parent DNA and a newly built copy. And so this is the completed parent strand going through the stages of DNA replication as it takes place in S phase. The first thing that happens is that helicase, an enzyme, is going to cut through the hydrogen bonds, break apart the hydrogen bonds that are holding together the nitrogen bases. So the blue, the green, the yellow, and the red are going to be split up through the action of helicase. You'll notice that first we need to figure out which side is the leading strand and which one is the lagging strand. The strand on the right, that parent strand, is going to be built in a continuous manner. So this new strand is going to be the one that I'm showing you here, is the leading strand. The purple piece that you see is an RNA backbone. So remember, primase has to initiate DNA replication because DNA polymerase III cannot do it on its own. So this little strand, or this little nucleotide here, is the RNA complement to the DNA complement on the other side. Notice too that that circular part of the purple backbone is consistent with the 5' end, while the parent strand was the 3' end. So we're building continuously in the direction of how helicase broke apart that DNA strand. And RNA nucleotide primers, there are usually 7 to 10 of those nucleotide bases, but in our model I just am building 3. Obviously, before DNA polymerase III can do its work, we're going to move over to the left side, which is now going to be referred to as our lagging strand. The lagging strand will be building in the opposite direction. So notice that yellow nucleotide is the RNA nucleotide complement to the DNA strand of the parent, and its purple end, that the circular part is the 5' end. So we're going to be building towards the screen, and so you can actually see that. So I built 3 little RNA nucleotide primers to represent the activity of primase. So now once we have primed both the leading and the lagging strand, DNA polymerase III can come on in and start building. And so you'll notice that the DNA nucleotides are, we're using the gray backbone piece to represent that it's a DNA nucleotide as opposed to an RNA nucleotide. And so this will continue on that lagging strand, and then it will continue on the leading strand as we're putting in those gray or DNA nucleotides here, and it will build continuously. So the side on the right is our leading strand, and the strand being built on the left is going to be our lagging strand. Notice how the 2 new products are producing 2 DNA strands, adult strands or mature strands, that are anti-parallel as well. This action will continue, so I'm opening up the strands again. We still need to prime the strands because, at least on the lagging side, the leading strand, as you can see, can continually build because there is a DNA nucleotide already present, so DNA polymerase III can continue its activity, but the lagging strand has to wait until helicase opens opens up and exposes more of those nucleotides, and then we can put in an RNA nucleotide primer. I'm only using one here because the segment is very short. Once that RNA primer, that RNA nucleotide primer has been placed, then DNA polymerase III can add in those DNA nucleotide complements. So notice here we have a lot of gray interspersed with some purple, and on the other side we also have a new mature strand, but it only has, sort of one little segment of primer. Before these DNA strands are ready to, be delivered to new cells, remember DNA polymerase I has to go in and splice out those RNA nucleotides and replace them with DNA nucleotide complements. This is accomplished. Once this is accomplished, you get two strands, both running anti-parallel, that are exact copies of each other, which demonstrates DNA replication as a semi-conservative mechanism that occurs during S phase of interphase, a precursor to mitosis and meiosis. I hope this was helpful.